Good morning, of you. Welcome to Young Science Leader Series. Today with us Dr. Rajni Garg. She is a DST Inspire faculty at JNCSR and MCB Department of the Indian Institute of Science, Bangalore. So she has earlier worked as at Amity University in Haryana, and she has got several awards and recognition, including DBT BioCare Scientist, SBT, Jawaharlal Nehru University, New Delhi, and she holds PhD in biotechnology from School of Biotechnology, JNU. And she has been recipient of many national awards, including AMI 2015 Young Women Scientist Organizers Award, DST Inspire Faculty Award, first prize in oral presentation in BioPoch 2013, ICMR and CSIR international travel grants. Current research specialization include molecular biology of infectious diseases like tuberculosis, where right now she is working in a BSL-3 facility of JNCSR and IISc Bangalore. So thank you so much, Dr. Rajni, and she is also actively contributing in our virtual internship. She has been already promised to guide few students. So in case some of you are interested to contact her, I welcome. So if you would like to work on infectious disease, there's a tremendous scope in this field post COVID-19. Yesterday also we were talking about it. So you can also ask. We are going to have extensive question and answer session today after her talk about the opportunities in infectious disease in particular and tuberculosis as well. So interested to pose any questions, you're welcome. And so today with me, this session is co-moderated by Miss Geetika Garg. She is also a very active participant in our Young Science Leader as well as the earlier workshop. And she posed one question also yesterday. And then I requested her to moderate this session. Why not? So she is basically doing MTech in marine biotechnology, a very, very unique. I've never came across any universities in India offering MTech in marine biotechnology. Of course, it's only one place. It's a DBT lab. The lab is known as National Center for Aquatic Animal Health in Kochi, Kerala. So if any of you is interested on MTech in marine biotechnology, she is here to share her experience. And of course, she qualified after something called JNU CEEB exam. That is also known as GAT-B exam. Over to Geetika Garg. So you can introduce yourself first, and then we will start the presentation.
Ma'am, Rajni Garg, ma'am, Felix sir, and all the young science leaders out there, a very hearty good morning. Myself, Geetika Kumari. I am currently pursuing my MTech in Marine Biotechnology from National Center of Aquatic Animal Health, Kochi, Kerala. It's a DBT-sponsored program, and I was admitted to this program after qualifying a national level exam of JNU CEEB, which is currently being taken and conducted as GAT-B exam. Now, coming to this platform, first of all, I would like to thank Felix sir. Felix sir, thank you so much for giving me this opportunity of introducing myself, introducing such great speakers, and even moderating this session. Thank you so much. Rajni Garg, ma'am, it's a huge opportunity. It's a privilege getting you here, knowing about your work, knowing about your journey of becoming such an outstanding science professional, ma'am. It's Thank you so much for being here, ma'am. Please continue with the presentation. Over to you, ma'am.
Thank you, Geetika. Thank you, Felix sir, for getting me here in YSL. So Felix sir, I would like to tell you that I first came across you at Indian Scientist Response to COVID-19. You are part of that group. And through that, I got to know about SLW and I registered for the workshop and really got benefited out of it. And then I got to know about YSL. So thank you for waiting for me while I was making a move from Bangalore to Chandigarh. He waited for me for my session so that I'm comfortable, settled in my new house, and then I can give a good talk. Thank you so much, sir, for giving me this opportunity. So, and I would like to share my life journey, and I hope if it is beneficial to even a single person, a single woman scientist, then I'll be really grateful. And I'll start sharing my screen. Is it visible?
Yes, go ahead, please.
Yes. So I have named my talk as Journey of a DU Girl, from a DU Girl to a Woman in Science, an Inspired Tale. This
Yeah, you can continue, Rajni.
it's not moving actually. Yeah. So I would start with my family and childhood. So I was born in a business family in Delhi. So at that time, my family was very simple, born in a joint family. So you can see the pictures we used to enjoy. It was a joint family. We used to enjoy gatherings, everything. And and I was always among the top three students in school, in class. So I was very studious from the beginning. So my parents, like, it was dream of my dad that I become a doctor, a medical doctor, and also my grandmother, because I was very good in studies. So at that time, it was like, if a child is good in studies, you would like to become him, make him a doctor. So that was the perception. And so I took medical, and then I continued with medical. So these are my trophies which I got in school itself. So my mother is really very fond of the trophies. She has put them in a showcase, and she always keeps on cleaning them and putting them nicely. So it was during my 11th standard when my biology teacher, she was herself a PhD. Her name is Dr. R.J. Goendi. So she told me about this National Children's Science Congress. So it is like a national event in which like, children are supposed to make small projects. So she said that you are good in biology, Rajni. So why not we try making a project and we go for it. So we took up a small study on diabetes mellitus. So we all know that Karela juice and fenugreek seeds, the indigenous herbs that we use, they have a role in diabetes. But at that time, there was no documented study on these. So what we did was we took a small cohort in our nearby school area, and we used to give them prescribed doses. We used to go to their houses and give them doses of Karela. We used to give them doses of fenugreek seeds and other herbs. And we used to monitor their glucose level by glucometer. So, and in 2 months time we could see their glucose level coming down. So it was really a good study as far as school children are concerned. So the project got selected at national level, and I was the team leader. That was basically my first experience of leadership, I will say. At a very early stage, I got this experience. So I was the team leader, and I got to present the work at Pune at National Children's Science Congress. And there, for the first time, I heard Dr. APJ Abdul Kalam and Dr. R.A. Mashelkar. They were encouraging students to take up science as a profession. I never thought of that before, because I thought that medical is the only thing that you can do if you like biology. So I was myself very scared of blood. So I thought that why not science? It's a good option, right? If you don't want to do medical, don't want to go into surgeries, becoming a doctor, becoming a professional medical doctor, then why not take up science? Because I used to love biology. So I gave my medical exams, but I didn't clear because I think I was not very keen on doing medical. And then I did very well in my board exams, and I got into a course called BSc Honors Microbiology course from Delhi University. So it was a really sought after course, and I thought that being a 90 percenter, I'll make it to it, and did make it to it, and it was taught in just 4 colleges at that time in Delhi University. At that time, I didn't know anything about microbes, but when once I got into the college, I realized what are microbes, And I used to spend hours in practicals. I used to love doing stainings, fungal sections we used to do, and I got really interested in microbiology. We got some really excellent teachers, and they were like, we were not, it was like they used to teach us, they used to do practicals, they were all the time with us. So I got excellent teachers, Dr. Sudha, Dr. Rakesh Kumar Gupta, Dr. Salomi Jones, and Dr. Kusum Gupta. So they taught us variety of subjects, biochemistry, microbiology. I was taught diversity in microbiology from algae to protozoans, everything we were taught extensively. And Dr. Kusum Gupta used to teach us immunology. And in DU, there's a culture of senior junior. So our seniors used to help us a lot with notes and everything. So I'm really thankful to all my seniors who helped me. And there used to be a lot of discussions like what next? What will we do next after BSc? So the immediate thing that came to our mind was MSc. And MSc has entrance exams. So we got really help from the seniors how to tackle entrance exams. And I made through entrances and I got admission in MSc Biomedical Sciences in ACBR. So ACBR is in heart of Delhi University. It is in North Campus. So I consciously went to do biomedical sciences because microbiology in itself was very restricted and specialized course. And I thought that if I want to do research as my career, then I should have exposure of other subjects also. So getting into ACBR, it broadened my thinking. I learned a lot of new subjects and I got really feel of a lab life, what happens in lab. So I realized that lab is not just doing experiments. It is about a lot of fun things also. And we had a lot of fun doing experiments. We took breaks also and we got proper DU feel in that. And I did my dissertation with Professor Daman Saluja. At that time, I worked on tumor suppressor genes. In my 6 months dissertation, I realized that studying biology is different, but conducting experiments and getting results, oh, it's really a tough task. You do experiments, they fail. You do again experiments, they fail. Then what to do? So in my dissertation, I didn't have any major results. So I went to ma'am and told ma'am, see, this is happening, this is failing, this is going like this. So ma'am said that, don't worry, Rajni, it's okay. Like, this is just your training. What will you do next? You think about that. This is just your lab training and it happens. It, that is why PhD takes a long time. Professor Daman Saluja was a director of the institute at that time. And I was really amazed by the way she used to handle administrative work as well as her lab. So it was really a privilege talking to her and getting trained by her. Another mentor which I got was Professor Vani Brahmachari. She used to work on epigenetics, Drosophila genetics, and such an amazing teacher she was. And she was from IISc and so humble. I really the poise with which she used to carry herself. I really look forward like getting mentors like that. I was really lucky. And with these things, I like made up my mind that okay, we'll see how it goes. Let's do PhD. Let's see how it goes. And in the meantime, like my family used to keep on talking. Being from a business family, I was the first one to do an MSc in biomedical sciences. So they used to ask me, what will you do next? What will you do next? When will you get married? What will you do next? So I told them that, I like teaching. So I like to teach also. So if I complete my PhD, then I can get lecturership in DU. So my dad was happy. Okay, let her do PhD. She will become a lecturer. So that will be a really nice career for her. She can manage her house also, and she can manage her this thing academics also. So with this thoughts, I went on to give PhD entrance exams, and I made it to Jawaharlal Nehru University in PhD biotechnology. So PhD was a turning point on my life, both at personal level as well as academic levels. So I joined Professor Rakesh Bhatnagar. So his major work is on vaccines. He had at that time many ongoing projects, and he had developed anthrax vaccine, and he also had projects on rabies vaccine. So they had developed rabies DNA vaccine. So vaccine that was used at that time for rabies, it was the rabies vaccine. It had lot of side effects and used to take lot of shots for that, for proper immunity buildup. So we were working for a rabies DNA vaccine. Okay, so one of my seniors in the lab, so she had already developed a rabies DNA vaccine, and that vaccine was showing a very good efficacy in mice model. So what my objectives were to test it with human approved adjuvants so that we can patent the vaccine and then we can make vaccine available for human use, and also to do the dog trials in rabies vaccine. So I started my PhD on this topic. But then things happened, and my PhD was really a roller coaster ride, because I met a boy called Anupam, and we liked each other, and we decided to tell our parents that we like each other and we like to take forward this relationship. But we didn't want to get married at that time. But if you tell your parents, then they want you to get married, right? Because they will say that get married and pursue your career later. So they wanted us to get married. And my PhD work, as I told you, I had this vaccine, but there was not enough things to explore. I wanted to do something more. My PhD work at that time was not very exciting. So I went to professor and I told him, sir, I want to do something else. I want to do something basic, which is more intriguing and something which I learned, explore more, in which I can explore more. So he asked me that in interview you told that you would like to work on tuberculosis. Do you still want to work on it? I said, yes, sir, definitely. So at that time, he had got a project for which he got a BSL-3 facility named. So that was basically for working on anthrax. But if you have a lab, BSL-3 lab, and you have two separate BSL-3 rooms, you can work on two microbes according to guidelines. So we then thought, why not work on Mycobacterium tuberculosis? So I worked on Mycobacterium tuberculosis with Dr. Nirupama Banerjee. Dr. Nirupama Banerjee was his wife in ICGB. So Sir said that they have really interesting project and they are looking for someone to take forward that. But it's really challenging because you'll have to standardize everything in BSL-3 lab. They have in ICGB a very good lab in which all the protocols are standardized. But our lab we are building here. And I thought it's a very good opportunity to go ahead with. So I learned the techniques in ICGB, all the protocols and everything. And I got the things here. And I started standardizing the protocols, standardizing animal experiments in our own BSL-3 facility, which is housed in School of Biotechnology. So this is the inside of BSL-3 facility. You can see a person working in PPE kit. So nowadays, this is a very common scenario because of COVID-19 situation. Earlier, back in 2010, no one used to no one used to see these pictures often. So this is inside of a BSL-3 facility. So all these things were going on. I changed my topic of my PhD, and my personal life also, like parents were saying, get married. So with all these things, I got married in third year of my PhD. But my PhD guide, Professor Bhatnagar, he told me one thing, that Rajni, you know why so many undergraduate, there are so many girls at undergraduate levels, but hardly any of them make to the faculty level, because girls yield to their family, they sacrifice their career for their family, for their spouses, for their children. I have full faith in you. You should never yield. So these were his words. So I told him, yes, sir, I will never yield. And I rolled up my sleeves to take on this journey. And I got married. And simultaneously, I carried forward this exciting project on TV. So I would like to share with you some highlights of my PhD project. I tried to make the slides simple because the audience is diverse that we have here. So, so I worked on a genetic locus RV0574C of Mycobacterium tuberculosis H37RV. So MTB has a very thick cell wall. It has variety of layers in the cell wall. So these layers are mycolic acid, peptidoglycan, and arabinogalactin. So beneath arabinogalactin, there's a layer called polyalpha L-glutamine layer. So very little is known about this layer. And this layer is responsible for giving MTB cell wall strength, which makes it impervious to many drugs. So previously in Dr. Nirupama's lab, what has happened was one of the seniors, he found that there's a protein called glutamine synthetase protein, which synthesizes glutamine. And this glutamine has a role in developing this polyalpha glutamine layer. But how this layer is polymerizing, whether this layer is intact in the cell, how is it, nothing was known. Because for a pathway, there are other proteins also that should take part. So they were looking for other proteins in this pathway. So through their bioinformatic analysis, they had found out that this locus, this RV0574C polyglutamate synthase like enzyme, this might have a role in synthesis. This might have a role in synthesis. So we went on to do experiments, and this particular RV0574C locus, it is a part of MTB dormancy regulon. So MTB has a 48 gene dormancy regulon which gets activated when it is inside the host, and it stays like near the expression level of these genes is very less in normal growth conditions. So in hypoxia, it increases, in acidic stress, it increases, even in nitric oxide and in carbon dioxide stresses, the level of this gene increases. Then I created a knockout for this gene in MTB, and we found out that the knockout has rarely less biofilm formation. So for MTB to colonize in the host, biofilms making is the very strong capacity. It is needed. So if a mutant is not able to form biofilm, it is considered to be attenuated. We also saw the cell wall strength of this. So since polyglutamine is known to have a role in cell wall strength, is our mutant deficient, being deficient in PGS, the RB0574C, is it contributing to cell wall strength decrease? So we plated the cultures on SDS as well as lysozyme, which are cell wall disruptants, and we found that the growth is less, and the mutant also showed fourfold lesser PLG formation, and it was more sensitive to SDS and lysozyme. The mutant was also attenuated for growth in macrophages. So these experiments were done in cell lines. They were infected with the wild type as well as the mutant and the complemented strain, and the complemented strain would restore back the phenotype. Also, for the cell wall strength experiments, we did this 50%, we found 50% decrease in viability by mechanical stresses like weed beating. Also, we did animal experiments, and we found out that, so this is lung section of mice which are infected with wild type MTB. So lungs have various free spaces which are called alveoli, and when there's an infection, there is infiltration by the immune cells. So you see that these spaces are filled by the immune cells. So this is wild type H37RV. This is the control, which is the mutant. And you see that the mutant has hardly invaded the, because mutant is cleared very fast, so it is not able to invade the lungs. And this is the complemented strain. Here again, you can see the infiltration by the immune cells. And this is control, in which you can see the alveolar spaces are intact. So I got to do this work and I was really fascinated about my work. And then another thing happened that I conceived at that time. So I was like in the last stages, I was writing my thesis, I was writing my paper, and I conceived. So I thought that phase, my pregnancy went fine. Everything was, during my pregnancy, I submitted my thesis. Everything was going on fine. But the thing was, when we communicated the paper was communicated to infection and immunity, and it came back like after 1 month my child was born. The paper came back with major revision. So I was thinking, I thought that having done so much, like it will go. We had thought, like my professors and I had thought, but then we didn't want to lose this good journal. And having done so much, I wanted my work to be published in a good journal. So I like major experiments were suggested, and I was in a fix, like what to do. So I took 1 month time, and I decided like how to chalk out a plan. So sir gave me full freedom, like you can chalk out your plan and let us know. We'll have a discussion when you are ready for a discussion. So I chalked out a plan. What I chalked out, I thought that being a mother, I have to, it is true that I have to take care of the baby, but my academic responsibilities are also there. So I have to do that also. So when manuscript revision came, I used to work on flexible timings, and I took help. I took help from my juniors. They are also co-authors on the paper. So I took help from my juniors. I used to spend hours on phone. I used to be like this, handling the baby and on the phone. I used to like make what all they can do, what all I can do, and I used to go whenever it is required. So after 2 months of my baby was born, I was back in lab, back at bench, and I completed major experiments with help of course, with help of my lab mates. I could finish it, and the paper it went through, and the paper again a major revision came. So it took me almost, like my baby was born in January, and the paper was accepted in October. So a whole lot of time I was like with the baby also and working also. So my family, Anupam's family, took a lot of care of the baby, and I used to go and used to come back. They used to like bathe the baby, sleep the baby, everything they did, because it was there in their mind that she also has to take care of her career. So I'm really thankful to my both families. They took care of the baby, and then I could take care of my work. And this we got this paper. The conserved hypothetical protein RV0574C is required for cell wall integrity, stress tolerance, and virulence of MTB. So it gave me back my confidence, which I had lost, that how will I manage both the things. And I was like, there was no looking back after that, because I had now make up my mind that I will continue in science, and I will not be like, I will not take up teaching alone. I would like to do research as well as teaching if I get an opportunity. So meanwhile, my husband, I missed out on that point. He didn't like the lab in which he was there in JMU, and he moved to PGI Chandigarh. So he was doing his PhD there. So there was some time, he started late because his time, he started late and he had some time in finishing. So I wrote a grant. I wrote my own grant, DBT Biocare. I wrote a grant for DBT Biocare. So this is a very good fellowship for women. So they encourage women with gap years also. So I am lucky. I have no gap in my CV till now. but this is for women who have gap. They give preference to them who have gap in their research career. So we worked on a development of diagnostic kit for detection of tuberculosis and drug resistance determination. So this work was done in collaboration with the Lalpath Labs. So tuberculosis, as we all know, earlier it was considered to be disease of the poor, but now we know with emergence of multi-drug resistant, drug-resistant strains, and as well as coincidence with HIV, it is no longer disease of the poor, but still the drug regimen is very long. 6 months to 2 years it takes to cure TB, and it has four highly toxic drugs are used. The problems faced are hepatotoxicity and non-compliance. So we thought that why not develop a diagnostic kit, a quick kit that can give result quickly rather than relying on gene expert. Okay, so gene expert was is a WHO recommended device which is used for tuberculosis diagnostics, and it also tells whether the disease is rifampicin resistant or not. So the cost of Gene Expert, one test of Gene Expert, which is done in one cartridge, was 1100 to 3000 rupees, and it was not available at resource constrained settings. So we developed a kit which consists of this. It detects MTB as well as NTM. So NTMs are non-tuberculous mycobacteria, which by acid fast stain and other techniques, they will look like mycobacteria, but they are non-tuberculous because the person will not have tuberculosis disease. But since there is no proper diagnostics, they will be mistreated. They will be given drugs without knowing that it is actually tuberculosis infection. So our test could diagnose differences between tuberculosis and NTM, as well as we did a sequencing reaction, which could sequence the rifampicin resistance region. And we can see whether it is rifampicin resistant or not. So this work was carried out with Lalpath Lab. So at our BSL-free facility, we developed a proof of concept with pure cultures that such a test is possible. And we got samples from Lalpath Lab, and and this was validated on those samples. And we got very good sensitivity and specificity, which was of the level of WHO recommended gene expert. So then we validated this, and this test Lalpath is still continuing by name of TB Sure. This comes by name of TB Sure. They are using this for routine diagnostics. Then after this, Anupam was over with his PhD, and we were at crossroads. Like we wanted to know, we wanted to see what to be done next, whether we should do our postdoc from India or abroad. So that was a big decision of my life. Uh, so we wanted to stay together. Having stayed apart for so long, we wanted to stay together, and we wanted our like child to have both of us together. So we were looking for positions. Then Anupam was very keen on staying in India. He thought that if we can do something for the country, being so educated, that will be great. We should. So our preference was in India, and he was very keen on staying in India. So I applied for some. I applied for fellowship opportunities in India itself. I had a reasonable good CV at that time. This was back in 2015. So I applied for this DST Inspire faculty scheme. So I got this DST Inspire, and again I was so much interested in tuberculosis. I applied for this tuberculosis grant. This was characterization of RB1997 CTPF of Mycobacterium tuberculosis H37RB. So I joined in Amity University first. So Amity University, I was there for 6 months, and I realized that I was 29 at that time. I realized that I do I really want to become a full faculty at this time? I wanted to, because there was so much to learn. I thought it will be better to go to an institute where I can learn more, because it's a vast ocean. Science is a vast ocean. Rather than being entirely on my own, if I get a mentor, how about it? Even in Inspire Faculty Grant, if I get to get a mentor who can give me, like, who can give me freedom for my own work, and I get to learn things from him also. So I was in this dilemma when Anupam got a very good postdoctoral opportunity in INSTEM Bangalore. So, and then I also applied in with Professor V. Nagaraja at GNC. He was GNC president at that time, and his lab was in microbiology and cell biology at IISc. So I wrote to him, and he happily agreed to mentor me. So we decided to move to Bangalore. My son was 2 years old at that time. So we decided to take a move. I shifted my grant there, and so Bangalore, it was a new lab, new life. So we thought that how will it work out? Because we knew that our labs are very demanding, lab schedules are demanding, and at back home in Delhi we had a lot of family support for the kid. Now we don't have. So I was like trying to figure out what to do, and Anupam was also figuring out how do we chalk out a plan. So I came across this book, Lean In, by Sheryl Sandberg. So Sheryl Sandberg is Facebook COO. And I was surprised the kind of thing that we Indian women face at work or at home. The scenario is not very different in US. It is faced globally throughout by all the women. They are all the time they have like home things also running in their mind. They have to take care of the child also, and academic responsibility, office responsibility. So taking cues from this lady's book, we chalked out a plan. Like, we thought that there will be 50% work division at home also. So we decided that I will cook breakfast and pack lunches for both of us, as well as Ansh, my son, and he will cook the dinner. So we looked for a dedicated daycare for my son. So NCBS, we were fortunate. We got a very good daycare facility. So we used to drop him at the daycare, and we used to, like, the time we could not make because we had to collect Ansh at 6:15, 6:30 from the daycare. So we used to half day, we used to work on Saturdays. Like, I used to go in the first half, Anupam used to go in the second half, because Saturdays daycare was only half day. And on holidays, we also used to do half days because our experiments were such that we cannot take holidays. We used to take just Sunday off. And in between all the holidays, apart from major festivals, we used to go to work, and we used to do half day. So with this protocol, so this was our home protocol, not lab protocol. This was our home protocol. And we set out for our new journey in a different city. And this is my mentor, Professor Nagaraja. And I learned a lot in his lab. So the lab is very well managed. So they have this shared reagent concept. So we make common reagents, like reagents for SDS page, reagents which are used routinely for protein purification. So we have lab stocks for that, and everyone has rotations to do to prepare lab stocks. And we have daily responsibilities, which are division. Week wise, there is division of labor. Like, this person is supposed to do this work, this person is supposed to take autoclave responsibilities, this person is supposed to do this arrangement. So in lab runs very efficiently if you have a good leader. So running a lab is a very different thing than studying or doing PhD also. So work atmosphere was very cordial, and we used to have IISc is lot of academics goes on in IISc, very different environment than JNU. So there are a lot of brainstorming discussions, seminars, meetings, and we used to have Monday morning. We used to have 8:30 meeting, 8:30 a.m. in Monday. And we used to have evening group meetings that used to last for 2:30 hours, 3 hours. We used to have lots of discussions. So I learned that science, we grow as we discuss things with our colleagues. We grow, we learn their perspective, and how can we move forward using all the perspectives putting together. So I worked on my own project, CTPF, and I got to work on a few other projects as well. So this is CIDR. So this was my new Karma Bhumi now. This is DSL3 facility of Indian Institute of Science. And I most of my time I used to spend here working on MTB. So for my own Inspire grant, I worked on CTPF. So CTPF is an integral membrane protein. So it was not easy to work with a membrane protein. I had no previous experience of working with a membrane protein, but as I told you, I like to do adventures. So this is 100 kilodalton protein. So the thing was, when we were trying to purify it, it was never in soluble form. So we chalked out a plan that why not we isolate the membrane vesicles if we want to show the activity, whether the protein is active or not. We enriched, this was an MTB protein. We took a surrogate host and we enriched CTPF in M. smegmatis in the membrane, and we isolated the membrane vesicles, and we did this experiment of ATPase. It's an ATPase. So we got to know that it exports calcium. So this was not known before, that what is the ion that this transporter exports. So it exports calcium. And we thought this is also a dormancy regulon gene. So dormancy regulon gene, as I mentioned, I worked on in my PhD also. And the dormancy regulon genes, they are overexpressed highly when the MTB bacteria is inside the host. So we thought that it is a calcium exporter. What is this exporter doing when it is overexpressed in the host? So we did this microscopy. So confocal microscopy, I learned extensively and used in IISc. So we found out that if we generate this knockout, conditional knockout of CTPF, conditional knockdown of CTPF, the levels of calcium are very low in the macrophages. So it means that when RV is inside the macrophages, it is exporting calcium in the macrophages by means of our exporter. And this is a chelator for the calcium, BAPTA. It is a chelator. So the signals were diminished, and we got to know that it is not exporting, that the signals are less, and it is actually calcium which is getting transported. So CTPF mediated calcium, it we got to know that we did experiments, and we got to know that this calcium mediated efflux help MTB in inhibiting autophagy. So autophagy is a self-clearing mechanism of the host by which it clears out the invading bacteria. But MTB, it has got its ways to inhibit autophagy. That is why it is able to thrive in the host for so long, and it is able to go to dormancy and live happily in the host. So I did this work. This work is my first corresponding author work, and it is under review in Frontiers in Cellular and Infection Microbiology. Also, I did work on nucleoid associated proteins. So, nucleoid associated proteins. Till recently, we used to think that the bacteria, there is no membrane, nucleus is poorly organized, there is no intact membrane as nuclear membrane as the eukaryotic cell has. But now things we are deciphering that from recent years we are deciphering that nucleoid itself is a highly organized structure in the bacteria. So there are this set of proteins called nucleoid associated proteins, and they help to compact the nucleoid to fit inside a 1 micron bacterial cell. So nucleoid associated proteins, they bridge the DNA molecules and they coat the DNA molecules. So these are the nucleoid associated protein. This is the DNA helix. And they have different modes in which they bind DNA molecules. And by different modes, they regulate gene expression in by different mechanisms. So they are eukaryotic counterparts of chromatin remodelers or histones. They are eukaryotic counterparts of histones, as nucleoid associated proteins are basic proteins. So in our lab, this very distinct nucleoid associated protein called RB3852, we worked on it. So this protein is distinct because on one hand, it binds to the nucleoid, and on the other hand, it tethers to the membrane. It tethers to the membrane, and that is why it has distinct role in physiology. So my other project which I was working on, that what is the role of this protein in MTB physiology. So I got to do really exciting work in finding out interacting partners of this protein. So since it is going to the membrane, it is interacting with various proteins in MTB membrane, and it is contributing to cell division. It has various mechanisms to go to cell division, and it also has influence on global gene regulation. So currently the work I'm pursuing, it has influenced this. We are uh, like addressing the case that how this protein is affecting global gene regulation and infection scenario in MTB infection scenario. So this is the ongoing work, and we are also preparing the manuscript for it. So this is my journey, and so now this is my husband Anupam. He got to do some fantastic work and at INSTEM Bangalore, and he's now a faculty in PGI Chandigarh. Last year he got a faculty. And this is myself. This is my son. He is now 6 years old and very naughty. And he does many experiments on his own now. So in lockdown, I make him do a lot of experiments. He actively contributes to them, and his this is showing how germs stick to the finger and how a soap solution can clear that out. So I told him to like to teach him about COVID, about Corona. I told him this to do, and he's very excited about it. He always washes hands. So, a few key mantras which I can give to young students who are like want to pursue research as a career, to young students as well as to women students, to girl students, that I think it is very important to be a happy scientist. You should take breaks, take coffee breaks, you should discuss with your colleagues. You know, lab environment as such, there are a lot of stresses, and if you go out for some time, even a 15-20 minute break, it rejuvenates me. And you should always have a hobby. So apart from being a scientist, I am a dancer, I am a painter as well. So whenever I'm, disheartened about anything, I get back to my hobbies, and when I feel rejuvenated, I come back and I do the experiment. So it is very important to choose mentors wisely. I know it is not always in your hand to choose your mentor, but you should follow your instinct. There are a lot of things which you can gauge about a person when you meet a person. So, and follow advices of seniors. That is very important. I have been fortunate. I got very good seniors, very good mentors. And you should ask questions, be adventurous, and you should always ask for help. Being women, I have always asked for help at night, like take my cultures out. So at times I used to feel I'm bothering people, but then I realized that what else I can do? I have to get my work done, right? So, and people, they are ready to help, but you have to ask for it. And there are certain quotes which I follow in my life. Lean In by Sheryl Sandberg. So Sheryl Sandberg, she says that as women, we confine ourselves. We ourselves get back, like, I am not good enough. This question, it always keeps on hovering your mind that you are not good enough. Even if you get an opportunity, you say, no, 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 I cannot do this. So what Cheryl says that why not lean in? Why not take up the opportunity and then see whether you can do it or not? At max, you will be a failure, but you will be happy that you took the opportunity and did your best. And my PhD mentor used to say, you should never yield your career for anyone. And my present mentor, Professor Nagaraja, VN, says, take control of the situation and not allow situation to take control of you. This is the mantra I used to follow in Bangalore. So we have completed 3.5 years of postdoc in Bangalore of this Inspire Fellowship. 4 years have completed now. So did extensive work, and this mantra, it came a lot of failures also, but then you learn from the failures and then you build up again. You research questions are such that you will always, failures will guide you to better experiment designing later. So I would like to thank CSIR for my PhD fellowship, DBT Biocare Award, Inspire Faculty Award, Professor Bhatnagar and lab members, Professor Nagaraja and lab members, and my both families also. So this is our big group with Professor Nagaraja. This is at his house. And with these words, I would like to thank you. And I would be happy to take questions.
Yeah, thanks, Rajni. You can exit the window. Yeah, perfect. Geetika here.
Yes, sir.
You can start.
Thank you so much, ma'am. It was really inspiring, and being a married woman myself, I feel really related to you. So let's go towards the question. The very first question is from Soumya Ranjan Satrusel. He is asking, ma'am, how to design drug therapeutics against antibiotic-resistant bacteria?
Okay. So there's a lot of work which is happening on drug therapeutics against antibiotic resistant bacteria. So designing drug therapeutics, you have to refer to a screen, a drug library. Okay, that is the mandate for designing drug therapeutics. And for resistant bacteria, first, the question is that how will you test it? For drug resistant bacteria, you need to have a multi-drug resistant strain with you. And working on that strain is very difficult. Like if you say you want to use MTB, drug resistant MTB in India, it is used in very few labs. So designing therapeutics for drug resistant is a difficult task, I understand. But you have to have a lot of bioinformatics done, and as well as lot of wet lab for that. And this is indeed a difficult question that people are trying to address, but there's no simple answer for it because there are various steps from library screening to then testing it in the lab. There are a lot of things.
Yeah. Hi.
The very
Hello. Yes. Rajni, it was really fantastic, Dr. Rajni. Your talk, I absolutely enjoyed it. And especially on your, the prioritization of family versus profession. And you spoke a lot of things about your early childhood and your journey towards IISc Bangalore. It's absolutely inspiring. I'm really happy to have you here with us today. And yeah, I like many things, especially on the influence of this Sheryl Sandberg's, Lean In book. And then I never actually read that book. Of course, I'll definitely have a look at that. I think she's working in the Facebook, right?
Yes,
On the
Facebook
top
CEO.
of Facebook.
Yes.
CEO, yeah. So yeah, and because you are from North and you have been working in South for quite some time, especially in the IISc Bangalore. So, and the moderator of today's session, Geetika, is also doing the same thing, from North Bombay to the South Kochi. So how will you relate, can you share something about the intercultural communication that you have observed? You might be having meeting so many people from different parts of the world also in IISc and JNCSR. So can you share something about it? Intercultural communication, why it matters?
Yeah, intercultural communication. Actually, I really like South India because of in North India, actually, I have been born and brought up in Delhi. So, what I feel, people say something and do something. And in South, you see that people are more what they say, they do, right? And they are like I have interacted with a lot of people, lot of Kannadiga people, and a lot of people from Tamil. So I feel like a lot of people from who migrate from north to south, they have issues of intercultural. But I never got that because my lab is like very diversified, and we used to have a very healthy interaction with all of them. And we used to like they used to get us Kannadiga dishes, and we used to enjoy idli sambar, and we made a niche in there, and I was like when I'm not formally like out of IISc, I'm still working there, and I will be sad to leave IISc because I like the academic atmosphere of IISc is really thrilling. Like if we want to pursue higher education and everything, so it is very good. IISc Bangalore, like they have really good facilities and everything.
Yeah, and I also like the, some of the kitchen experiment. I'm a big fan of something called Naked Science podcast of the Welcome Trust and the Cambridge University. Every week I listen to it while walking in my
Okay.
park, and it also have a lot of kitchen science. And I saw that you have actually shown your growing up your young boy, the son,
Yes.
with the hand washing and why COVID-19 hand wash is very important. I like that one. That idea is really really interesting. That kind of kitchen science, somehow the projects are missing in so maybe we need to start this kind of we have to actually what should I say what it's kind of we should bring the science to home because the
Yes.
public trust in science is actually increasing day by day. I'd say. Yeah, that effort is really appreciable. Yeah.
Yes, I
And
agree with you, Dr.
yeah,
Felix. Yes.
Ridul, Ridul asking, ma'am, how can one judge the environment of a lab before joining the
Sir,
PhD or postdoc?
your voice is breaking.
Okay, wait, pause the question. Can you hear me?
So your voice is breaking.
Yeah, Ridul Trimmer is
Um,
saying, ma'am,
Not audible.
Geetika, go ahead. You ask the question.
Yes, sir. Yes, sir. Ma'am, Mridul Kamal is asking, um, ma'am, how can one judge the environment of a lab before joining for PhD or postdoc? Is it fine to quit a lab while doing PhD because people around you isn't really caring about you?
I think you should always go and visit the lab and talk to people. It is very important to talk to people to gauge what is going on. And you can talk to them off the record, like not in the lab per se. You can like ask them to come for tea with you, and then you can ask specific questions about the mentor. Because not many labs, there are good labs also, and there are bad labs also. We all know that. And this thing that the general perception about the lab, about how a person like is thought about by by the society or by the department is very important. Like how, and many people have different aspirations during PhD. Some people want very good papers. Some people want okay papers, but they want that their lab should have all the resources. Some people want a really, a very uh, intelligent guide, and some people want, okay, it does not matter if the guide doesn't guide me, I can do my things on my own. So people have different priorities, and according to that they should like venture out and find out about and take proper decision. Take time with your decision and consult as many people as possible.
So the takeaway message I get is you have to know your mentors very well. You have
Yes,
to go there, you have to talk to people, and
yes,
then go for taking your PhD.
yes.
Okay, thank you so much, ma'am. The next question is from Rekha Kokanti. It's a technical question. Does RV382 works alone or in association with other interactive proteins in global gene regulation?
So we are trying to address this question, and global gene regulation it is able to do on its own, but it has got interacting partners which is helping it to do other functions in the MTP cell, which we are exploring.
The next question is from Soumya Ranjan Satrusel. It's the same question even I will have. Ma'am, your family supported you wholeheartedly, and that's excellent. But what about the woman who doesn't get such support? What will be your supportive message for them?
What I feel is that women should always assert themselves. There are so many things which are there in our heart, but we are not able to talk about them, right? So we should be able to assert ourselves that our career is also important. And if you are not able to assert yourself at home, who will listen to you at workplace? This is, if your near and dear ones are not able to listen to you, who will listen to you at workplace, or who will listen to you at leadership positions? You should first make, leadership starts at home. You should first make your near and dear ones understand what are your priorities, and how to chalk out, sit together with your family and chalk out how will you address these questions, like follow a scientific protocol at home also, like, and you should make them understand, like, you should assert yourself, because we are not saying, that is why people are not listening. The more we say, the more people will listen.
Next question is from Kirti Rani Sharma. Is nucleocapsid protein of viral responsible for the invasion and anchoring of viruses into host cell, and how helped into spinning up multiplication of nuclear machinery into host cells?
This question is not clear to me. First is nucleocapsid
Uh,
protein. So
yes.
nucleocapsid protein
Is
does has attachment proteins which helps to invade, but um, what happens is it helps in invasion only the DNA or the RNA material of the virus get inside. Basic virology says that, right? What was the other part of the question?
um, is nucleocapsid protein of viral responsible for invasion and anchoring of virus into host cell, and how help into spinning up multiplication? How does it help in spinning up multiplication of nuclear machinery into the host cells?
So only the DNA goes inside, and viruses have this property that they can, use host DNA polymerases and RNA polymerases for their own replication. So capsid proteins just help in invasion, and only the viral nucleic acid material, the DNA or the RNA goes inside, and then they use host machinery, host proteins to replicate and produce their own products, to produce their own proteins.
The very next question is from Rekha Kokanti. She is asking, what is the advantage of using drug resistant detection kit over gene expression analysis against MTB?
So the advantage is the cost only, Rekha. So this Gene Expert cartridge was very expensive. So we thought that, and not all the hospitals and not all the civil hospitals, and it is not available to people all throughout, right? In India, there are many places resource constrained settings are there. So we thought of developing this kit, which the cost is very less. It is around 300 rupees, the cost. The functions are the same. The Gene Expert also detects tuberculosis and NTMs, and difference between NTMs and the rifampicin resistance. So ours is the cost difference only, nothing else. The concept is also different. We use a multiplex real-time PCR, and they have their own technology which they do not share, which is patented, which they do not share. But the probes and all they do not share. So we have our own set of probes, but the main difference is cost.
The next question is from Ravi Kumar. What's the current scenario over cholesterol-resistant bacterial pathogens?
Current scenario of colicin.
What's the current scenario over cholesterol-resistant bacterial pathogens?
Over colicin resistant. I didn't understand it actually. I'll skip this
कोई
question.
बात नहीं. Maybe ma'am will answer it later. We'll go to the next question. It's from Shushrut Shubham. He's asking, how did you choose your guide and labs in which you have to work? Same question.
So you get to choose what you have in your plate, right? What will you have? So whatever options I had. So in my PhD, I had options from NBRC, National Brain Research Center. I had options from JNU, School of Biotechnology. I had options from School of Life Sciences, JNU. So these three options were there with me. So NBRC I chose out because I was not very much interested in brain research. I was interested in more on in microbiology aspects. School of Life Sciences, my rank was not very good. I was not getting the lab of my choice. So I preferred School of Biotechnology because I was the first ranker and I was given the lab of my choice. So I preferred Professor Rakesh Bhatnagar because I had heard a lot about him. I had heard that his work is good. He's working on vaccines. And that is why I chose him. And I asked people about the lab, about his nature. So everyone was saying he's a very good scientist as well as a human being. So that is why I chose.
Ma'am, the very next question I wish to ask, how do you go for selecting molecular markers? Like
Gitika, your voice is not coming. Are you muted?
hello.
Yeah, now it's fine. Hmm.
Ma'am, I wanted to ask, how do you select the molecular markers for making your discovery, like CTPF, associated protein? How do you go for finding it? That this is the
So
thing which has to be found.
So this is like a really, this question comes to my mind also. Like if I have to write a new project, how will I choose? How will I be
Yes.
sure that this is it and not anything else? So the question. So I read lot of literature. So you should always pick up a protein which is like which is multiple literature say it is doing the same thing. So I picked up CTPF because there are a lot of reports which were saying that CTPF is upregulated in host-pathogen interactions. There were at least 3, 4 reports from different groups
Okay.
that was saying that CTPF is upregulated in host-pathogen interactions. That is why I chose that. Also, again, it's a gamble. So it might work out the way you want it to, or it not, and that is the fun of science. We cannot, you know, run away from it. So you have to do extensive literature research, okay, and follow your instinct because you have now read enough to propose your own projects, right? So
Yes, ma'am.
extensive literature research and your own instinct is what I will say to do forward.
And what if, ma'am, if I am currently going through literature, I've come up with a molecule that I'll work with this molecule. And I've started the work, and after some time, I see some other papers already being published on it. Then what should
Okay,
I do?
so one thing I didn't share with you. So the CTPA work is was a 5-year project. So last year in December, when we were like finalizing the manuscript, so 2 other groups they published the paper. So approximately 30-40% of the work was done. So we had to do more work to because in India it takes time to do experiments, and especially in MTB field it takes a lot of time. So we had to do more work to support our data, and we that was all in vitro work. So whatever I showed with the macrophages and the animal experiments, I did more extensive experiments and then I showed it. So it will always happen. You will say, why am I, why did it happen to me? But you cannot help it. People are doing work fast, and in abroad, doing science in India is different from working abroad. And you have to, being an Indian scientist, you have to be prepared for this. So you have to roll up your sleeves and work hard and at very fast pace.
Okay,
Thank you,
so I
ma'am.
got disconnected, for quite some time. There is a major storm on the way here in Bathinda.
Okay, okay, yeah.
I will pick up one more question here. Priyanka Patial is asking, Inspire faculties, in spite of their good work, are not getting absorbed. After spending 5 years in this fellowship, they are on the road. So what is your take on this, seeing the present condition of the Inspire faculty? So I think we discussed this matter just before this meeting. So yeah, you
Yeah.
have any thoughts on this? Fine.
So I would like to say, Felix, sir, that it is this will consider the job scenario of the country. We have very few jobs, right? So government supported us for 5 years and to make us eligible for assistant professor job. So I think I won't blame the government, and especially now the COVID-19. So I don't see that recruitment processes are on halt now. And I don't, these are the negative factors of staying in a country like ours. But I think the good work will prevail, and of course we will get absorbed. I am hopeful of that. That all the people who have done good work, who have will be absorbed in future, because DST is sending out letters to all the institutes, and they are sending out letters that we are potential faculty candidates, and they are telling them to seek our potent in getting absorbed as a faculty. So I'm hopeful that soon everyone will get absorbed, and we have to bear with the situation with COVID-19. So let's hope for the best.
I can also add on, Priyanka, the one who asked the question. So instead of simply targeting the top-notch institute like IISc Bangalore or IIT or IISer, you can also consider looking at the newly established central universities. We have excellent research facilities, and I can say my own experience here in our Central University of Punjab, where I also joined, just like the Rajni. I was also a DST Inspire faculty awardee here, and yes, all the faculties, whoever got this one, the DST Inspire or Ramanujam or Ramalinga Swami, are all permanent in our university. So there is actually a good trait, I would say, that the university is prioritizing making these prestigious fellowship holders absorb into our permanent. So yeah, you have a lot of new central universities in India. You apply there instead of thinking only IITs and IISer, or the national lab. So universities are also fantastic places now, because there is not much a cash crunch in central universities comparing with the state universities. So you may please consider applying in CUs as well. Yeah, Geetika?
Yes, sir. Can I take the next question?
Yeah, go ahead. I think we can sum up
Hmm.
after one or two questions, max.
Okay. Okay. Ma'am, the next question is from Soumya Ranjan Satrusar again, and he is asking how postdoc helps in getting good position at an institute.
At an institute, any institute. So
How does postdoc helps in getting good position at an institute?
postdoc is very important for a person to get absorbed as a faculty, because in postdoc you get to work on your own projects, you get to publish. Because in India, the kind of CVs, because I am in touch with my mentors, so they are in recruiting committees. They say there are a lot of good CVs. People have done so much work, so much nature, cell science people are publishing, and they want to get absorbed in India. So of course, postdoc publications are very, very important for getting absorbed in our country, because as such, the number of jobs are less. So obviously, you will be judged on the basis of your CV, which is the only thing that the recruiting committee can see before they call you for an interview. So yes, postdoc papers, publications are very important for getting absorbed. in getting a position in an institute.
Yeah, so I can also ask because you're working with the tuberculosis. There are several reports now coming that the BCG vaccine, that is basically for the tuberculosis, is kind of preventing or limiting the COVID death.
Mm.
So what is your take on this? Because COVID-19, of course, is a respiratory, it's airborne disease. That is what the WHO is now saying. So does the BCG vaccine help to decrease the death of the COVID-19?
Yes, Felix sir, actually, not just BCG. Now I was reading another article that came up, and that lady from US, she has tested BCG as well as pneumonia vaccine, as well as other vaccines, and they all have a protective effect in COVID-19 situation. So I think that COVID-19 human body has never seen before, right? So this is an entirely new thing, and there is some cross, in viruses there is some cross reactivity and everything because of the peculiarity of the virus capsid, and respiratory viruses, they have these attachment proteins, and tuberculosis also being a respiratory this thing. So the cell wall, and there is some cross reactivity which might give protection, I'm 100% sure. And it is not just with BCG, it is happening with pneumonia and other respiratory disease vaccines also. So I think these vaccines will be preventive. At least 50% efficacy, I expect them to give. Yes.
Yeah, fantastic. So I was just relating with a question from Bablin Kaur. Her question is that as COVID-19 is also a respiratory disease, so can we flip the receptors from outer side of the cells to inner cytoplasmic site so that coronavirus doesn't able to interact with the cells? Can we flip the receptors from the outer side of cells to inner cytoplasmic site? Which cell is not specified, but yeah.
So I think what she is referring to, what's her name? This girl, it's
Bablin Kaur.
Bableen. It is not very easy to flip the receptors because I have worked with membrane proteins. So the membrane proteins, they are integral. These receptors are integral membrane proteins, right? So they have a fixed conformation, and once the ligand binds, then only their conformation changes. So it is not possible to put the cytoplasmic side on the inner site and the inner side to the cytoplasmic site. It is just not possible. I don't think so. There's anything that can be done in the host cell, that per se she's saying. So we cannot engineer such things, and I don't see any. No.
Yes, Geetika?
So that was the last question, I guess. Yeah. Um, so thank you so much, ma'am. It was
Thank
a
you.
very, very wonderful session for us. How the way you managed everything, starting right from your personal life, again flipping back to professional life, even how your family members have supported you, that means a lot.
That
And
is really my family support. Yes. Mm.
touch wood, it should stay like it should
Yes.
always stay like this for every woman who is thinking of getting a good profession like this. And the way you also continued with your hobby, the way you're choosing your things, how to maintain a proper balance whenever you feel depressed. Even your thing, your I guess thesis got revised twice. It was
Paper.
still you oh, your paper got revised twice, and still how you maintained that balance between your work. So that is really inspiring, ma'am. Thank you so much for coming here and joining us and helping us. Thank you.
Thank you so much. As a woman
Yes, Dr.
scientist,
Rajni.
yeah.
Yes, it's amazing, Rajni. Your talk was absolutely inspiring, and especially you stressed on the importance of the happiness. Finding our own
Yes.
whatever the
Yes.
hobby that we like it. For example,
Yes.
I love I'm just picking up the violin and listening to podcast and walking. I'm also a cyclist. Doing the the gardening and composting, so many lots of hobbies. So I mean, things that makes you happy, that we have to actually do that. That should be our inspiration to push our professional life forward. So I think that message is really, really interesting. And also today I'm really happy that to have both of you in the panel, and because
Thank you
both of
so
you
much.
are married, have children, and still pursuing first-class science research. So I'm really happy and lots of respect. And please continue this spirit way forward. And let both of you be an inspiration for millions of watchers. Definitely they will be watching. A lot more people will be watching this video. And yes, women in science is very important. And yes, you are the future leaders of the country. Thank you so much, Dr. Rajni.
Thank you, Felix sir. Thank you, Gitika, for giving me this opportunity. I would like to stress once upon for the girls who want to pursue science that assert yourself. Your families will support you if you are strong enough to assert yourself. You should not hold back because its leadership starts at home. You have to tell your parents what you want to do. You have to tell your in-laws what you want to do. So please, these are Cheryl's words, assert yourself. So I'm reiterating that. Thank you so much. Thank you.
Thank you,
Yeah,
ma'am. Thank you.
thank
Thank
you,
you so
Dr.
much.
Geetika. Geetika. And let me introduce the next speaker. So she's Dr. Tozama Kibani, and she's from South Africa, and she herself is a very well-known figure in the world, in the field of ethnomedicine. So her talk is scheduled at 4:30 p.m. today. So join with a cup of tea. Goodbye for now.